Next-generation sequencing method. Use in whole genome sequencing. To sequence entire genome for complete DNA sequences, and whole exome sequencing. To sequence all exons in genome. And target sequencing. To sequence specific genes. And RNA sequencing. Or transcriptome sequencing. To sequence all RNA for gene expression profile. And single-cell sequencing. To sequence single cell to understand biological processes. And epigenetic sequencing. To examine modifications that affect gene activity. Without altering DNA sequencing. As DNA methylation. And proteomic sequencing. To enable precise protein quantification. And highly multiplex sequencing. To process multiple samples in single run. And chip sequencing. To investigate protein-DNA interaction. So, next-generation sequencing method. Initiate by DNA or RNA extraction. And library preparation. In process of fragmentation by fragment DNA or RNA into shorter sequences. And ligate adapters in both ends of fragments in process of indexing or barcoding. As mentioned in next slides. And cluster generation. By bind fragments to flow cell. And undergo bridge amplification to form clusters. For sequencing by using massively parallel sequencing platforms. And data analysis. By align output sequence to reference sequence. As mentioned in next slides.